Good afternoon. First of all, I would like to sincerely thank you for your kind invitation to participate in this prestigious meeting. In this speech, I would like to summarize the main studies developed in monocytes as key players in the development of thrombosis and atherosclerosis in antiphospholipid syndrome patients. It has been widely demonstrated that patients with antiphospholipid syndrome have a large incidence of vascular damage involving thrombosis, accelerated atherosclerosis, stroke, or myocardial infarctions, among others. Several mechanisms have been proposed to explain the thrombotic and pro-inflammatory tendency of these patients, mainly comprising synergic effects of autoantibodies with prothrombotic molecules, inflammatory mediators, oxidative stress markers, and a plethora of intracellular signaling molecules in immune cells. Numerous studies have shown that the pathogenesis of both atherosclerosis and thrombosis in APS patients involves many immune system cell types, among which monocytes have demonstrated to play a key role. These cells are, however, an heterogeneous cell type with divergent functions in immunity, atherogenesis, and reparative processes. Thus, in parallel to in the relevant role in innate immunity, monocytes can promote detrimental effects by modulating a lipid metabolism, secreting inflammatory cytokines and chemokines, and producing reactive oxygen species that drive the pathogenesis. Moreover, they produce tissue factor and proteases that contribute to thrombosis and atherosclerotic plaque rupture. In this way, several studies in patients with antiphospholipid syndrome have demonstrated multiple alterations in the activation status and secretory functions of circulating and tissue monocytes associated with the development of thrombosis. Thus, in the expression of tissue factor, the main inductor of blood coagulation in vivo, was firstly described by our group to be significantly elevated in monocytes from APS patients with a previous history of thrombosis, and related to the presence of high titers of antiphospholipid antibodies. Thereafter, the molecular mechanisms underlying the enhanced expression of tissue factor and the activation of monocytes in antiphospholipid syndrome patients was delineated by several studies. Thus, it was demonstrated that antiphospholipid antibodies induce the expression of tissue factor through the activation of MAP kinases and the nuclear translocation and activation of NF-kappa B in monocytes, in a process that is mediated by protease activating receptors PAR1 and PAR2, also expressed in APS monocytes. It was also demonstrated that there is a close relationship between tissue factor and vascular endothelial growth factor, so that VEGF might act as a regulatory factor in APL-mediated monocytes activation and tissue factor expression, thereby contributing to the pro-inflammatory and prothrombotic phenotype of these patients. Proteomic studies further demonstrated that antiphospholipid antibodies are also responsible for the altered protein profile of APS monocytes, involving the regulated expression of annexins 1 and 2, Rho A proteins, heat shock protein 60, and protein disulfide isomerase, among others. These proteins have been shown to be associated with the induction of a procoagulant state, as well as autoimmune-related responses. That general scheme was some years later completed by identifying the presence of a pro-oxidative status in monocytes from APS patients. Thus, we showed that antiphospholipid antibodies further promoted an increased production of reactive oxygen species by monocytes, accompanied by significant losses in mitochondrial membrane potential, which disturbed the redox status and were related to the overexpression of both prothrombotic and pro-inflammatory mediators in APS patients. These pathologic alterations were then reversed by in vitro treatment with coenzyme Q10. Recent advances in gene expression analysis allowed to perform broad-based gene expression profiling. In a recent study, Ripoll and colleagues analyzed the in vitro effects of antiphospholipid antibodies from APS patients either with pregnancy morbidity or vascular thrombosis on monocytes in order to identify different molecular pathways related to the pathogenesis of different clinical APS subtypes. Thus, genes related to cell response stress, MAP kinase signaling modulation, and cell interactions were induced by antiphospholipid antibodies isolated from patients with vascular thrombosis. In contrast, genes associated with cell adhesion, extracellular matrix, and embryonic and skeletal development were modulated by antiphospholipid antibodies purified from patients with pregnancy morbidity, thus suggesting that the autoantibodies from different clinical subtypes of antiphospholipid syndrome patients induced disease-specific genome profiles in monocytes. Using microarray technology, then our group identified in vivo shared and differential genetic patterns related to atherosclerosis and cardiovascular disease in primary antiphospholipid syndrome plus lupus, and lupus patients. The study was developed in monocytes purified from 61 healthy donors, 41 patients with primary antiphospholipid syndrome, 54 patients with lupus, and 31 patients with both pathologies. Microarray analysis showed an altered expression of a differential number of genes among monocytes from patients with each pathology, so that only 38 genes were found to be simultaneously altered in the 3 diseases, indicating the heterogeneity of each of these autoimmune disorders. Functional analysis of altered genes in the 3 pathologies showed that at least 30% were directly related to inflammation, atherosclerosis, and cardiovascular disease. Looking deeper into the functional classification of the altered genes in the 3 pathologies, we observed that there was a specific profile or signature of altered genes when we compare the 3 diseases, and that they were associated with the altered expression of various cytokines, chemokines, and receptors, molecules involved in angiogenesis, and molecules associated with lipid metabolism. Similarly, we also observed a specific pattern of altered genes related to oxidative stress and mitochondrial function that form specific interaction pathways when comparing the 3 diseases with each other. This specific alteration seemed to be more relevant in patients with primary antiphospholipid syndrome. Taken together, these results indicate that the gene expression profile allows the segregation of these patients with a specific characteristic that explain the pro-atherosclerotic, prothrombotic, and inflammatory changes observed in each autoimmune disorder. Current genome-wide studies have shown that the human genome is extensively transcribed and produces many thousands of regulatory non-protein coding RNAs. Among them, macroRNAs are small non-coding RNAs ubiquitously expressed with a profound influence in the regulation of almost every cellular process investigated. In humans, more than 2,500 microRNAs have been described so far, and the number is still increasing. MicroRNAs have been demonstrated to control a wide range of physiological functions, such as embryogenesis, cellular differentiation, proliferation, cytokine production, and apoptosis. Furthermore, their altered levels have been associated to a number of pathophysiological processes, such as cancer, cardiovascular disease, or immune-related diseases, among others. As for their mechanism of action, they bind by sequence complementarity to the promoter of the messenger RNA of their target gene, promoting its degradation or inhibiting its translation into protein, so that an increase in a given microRNA will imply a decrease in levels of its target protein, and vice versa. The microRNA reduction implies an increase in its target proteins. The first study that intended to characterize the role of microRNAs in the pathogenesis of antiphospholipid syndrome was published in 2011. That study identified 2 microRNAs as main regulators of the expression of tissue factor. Thus, the expression levels of microRNAs 19B and 20A in monocytes from APS and lupus patients were found significantly reduced when compared with healthy donors, and negatively correlated with the increased expression of tissue factor in the cell surface of monocytes from both pathologies. These studies suggested for the first time the potential role of those microRNAs in the pathogenesis of thrombosis in antiphospholipid syndrome patients. In a later study, we identified and characterized a number of microRNAs related to the cardiovascular disease present in 2 cohorts of APS and lupus patients. The selected microRNAs were recognized on in silico studies. They were main regulators of targets involved in clinical features of antiphospholipid syndrome, such as atherosclerosis, thrombosis, immune response, and oxidative stress. In monocytes isolated from APS and lupus patients, a reduction in levels of microRNAs 124 and 125 was found, while levels of microRNAs 155 and 146 appear increased. Furthermore, the altered levels of these microRNAs correlated with markers of thrombosis, inflammation, and oxidative stress, and were associated to the presence of thrombotic events, as well as with an increased carotid intermediate media thickness as a mark of early atherosclerosis. In vitro, monocytes treatment with antiphospholipid antibodies promoted the same deregulation of microRNAs observed in vivo. Taken together, this result established that specific microRNAs might be considered potential biomarkers of immune activation and atherothrombotic development in APS patients. Antiphospholipid antibodies are directly involved in the deregulated expression of these microRNAs. All these studies have evidence that there have been many advances in the understanding of the molecular basis of thrombosis development in antiphospholipid syndrome. Nevertheless, further research is still needed in many fields, particularly in the study of the relationship among changes in global transcriptomic profiles in monocytes with the positivity of autoantibodies and their involvement in different clinical manifestations. With these premises, we have developed a new study on which an integrated approach was employed by evaluating jointly data obtained by multiple gene and microRNA expression profiling methods in monocytes from APS patients in order to characterize distinctive clinical APS phenotypes and identify novel molecular regulatory networks related to cardiovascular disease. In this study, 40 consecutive patients fulfilled the classification criteria for primary antiphospholipid syndrome, and 40 healthy donors were recruited. Clinical and analytical profiles of APS patients were evaluated, including the analysis of microvascular endothelial function, the carotid intermediate media thickness, and the score of thrombotic risk, GAPS. In parallel, we performed microRNA studies to characterize the whole transcriptomic profiles of monocytes. All the data obtained was then integrated by using machine learning approaches, as detailed later. The study of the gene profile in monocytes allowed us to identify 547 altered genes in monocytes from patients with primary antiphospholipid syndrome in comparison with healthy donors. In the same exploratory cohort, we analyzed the microRNA profile in monocytes by using nanostring technologies. The results of this analysis show that 22 microRNAs were found differentially expressed in monocytes, including 3 upregulated and 19 downregulated. Functional classification of those altered genes and microRNAs showed an enrichment in functions linked to development of cardiovascular and connective tissue disorders. By using the microRNA target filter function with the Ingenuity Pathway software, we identify predicted or validated interactions between 19 microRNAs and 115 messenger RNAs. In this picture, it is highlighted the specific network integrated by those genes which have an overall impact in cardiovascular disease and their potential regulatory microRNAs. Then we wanted to further evaluate whether said molecular signature would be capable of clustering patients clinically into distinctive profiles according to their cardiovascular risk. By using K-means clustering analysis in the APS cohort, 3 clusters were distinguished, representing different molecular profile groups with respect to the monocyte expression levels of the signature integrated by genes and microRNAs. Additionally, partial less square discriminant analysis confirmed a clear separation between these molecular clusters, where some genes and microRNAs were identified as important variables that contribute to this discrimination. Then the clinical and laboratory profiles of each cluster were evaluated. Consistently, cluster 1 was described by patients that had suffered thrombotic episodes with low incidence of obstetric morbidity, high positivity for antiphospholipid antibodies, and high prevalence of cardiovascular risk factors. On the contrary, cluster 3 comprised the youngest patients with a high incidence of thrombosis and obstetric complications, but low positivity for antiphospholipid antibodies and the lowest prevalence of cardiovascular risk factors. Cluster 2 represented an intermediate clinical phenotype with thrombotic events and obstetric morbidity, and also intermediate positivity for antiphospholipid antibodies and medium prevalence of cardiovascular risk factors. Therefore, when comparing gaps between different clusters, we found prevalence of patients with a high or moderate gaps in cluster 1, while in clusters 2 and 3, more than 70% of patients display an intermediate or low AGAPS score. Moreover, those patients with the highest AGAPS score further display the highest incidence of atheroma plaques. Taken together, our whole data suggested that specific transcriptome regulatory network in monocytes seem to regulate the biological processes related to the pathology of antiphospholipid syndrome. The integrated transcriptome profile allow the identification of central genes, microRNAs, and pathways altered in monocytes of patients with antiphospholipid syndrome, particularly relevant to unique characteristic of the disease and at least partially regulated by antiphospholipid antibodies. As more insight is gained about the pathophysiology of antiphospholipid syndrome and the involvement of receptors, intracellular pathways, and genetic and epigenetic alterations, new treatment modalities have been proposed on which the patient's cellular and molecular profiles are becoming more and more relevant. Thus, in past few years, several potential new therapeutic approaches to APS have emerged, including combination of antioxidants, antimalarials, statins, and B-cell inhibitors, among others. Nevertheless, to accurately define the mechanisms underlying the response or no response of these patients to these therapies, the alterations that happen in such a complex disease cannot be understood as an independent units. Thus, the interaction between these molecular alterations must be evaluated to guide therapeutic decisions. In that way, in a previous study, we evaluated the global effects of fluvastatin on the prothrombotic tendency of monocytes of APS patients. 42 APS patients with thrombosis and 35 healthy donors were included in the study. APS patients received 20 milligrams of fluvastatin per day during 1 month. Blood samples were obtained before the start, at the end of treatment, and 2 months after the end of treatment. It is important to underline that during fluvastatin treatment, patients were not withdrawn from their therapy, so that fluvastatin was always given as an additional treatment. After 1 month of treatment, monocytes of APS patients showed a significant inhibition of tissue factor, protease activated receptors 1 and 2, vascular endothelial growth factor, and FLT1 expression, all of them related to the inhibition of P38 MAP kinases and nuclear factor kappa B binding activity. Proteomic analysis also showed reversed expression of proteins involved in thrombotic development after fluvastatin administration. In conclusion, the data from this study support the belief that fluvastatin has multiple profound effects in monocyte activity, which might contribute to the thrombosis prevention in APS patients. More recently, novel and specific transcriptome regulatory networks in APS along with inflammatory and prothrombotic profiles modified by effects of ubiquinol treatment, have been identified through the implementation of a prospective randomized double-masked placebo-controlled study. 36 patients with the APS were randomized to receive the reduced form of coenzyme Q10 ubiquinol at a dose of 200 milligrams per day or placebo for 1 month. Treatments were given according to a randomized and crossover design with washout periods of 4 weeks. During this treatment, patients were not withdrawn from their standard therapy. Changes in several parameters related to inflammation, oxidative stress, endothelial activity, mitochondrial function, atherosclerosis, molecular signaling, and mitosis, along with regulatory microRNAs were assessed. The global results demonstrated the high potential of ubiquinol to modulate markers associated with thrombosis and cardiovascular disease in APS patients, highlighting its role as a safe adjunct to standard therapies in this autoimmune disorder. Because of the notable absence of clinically significant side effects and its potential therapeutic benefits, this study suggested that ubiquinol might act as a safe adjunct to standard therapy in patients with antiphospholipid syndrome. Overall, all these studies suggested that the extensive molecular profiling of monocytes in patients with primary antiphospholipid syndrome might help to identify distinct clinical phenotypes, which in turn might facilitate a patient-tailored treatment. Although further wider studies are required to definitively demonstrate that coordinated deregulation in APS patients' monocytes, and even when other molecular actors might influence these alterations, these achievements in our understanding of the disease have opened the door to the possibility of new model-targeted therapeutic options for the prevention of atherothrombotic events in APS patients. Finally, I want to present here our research team. Thank you very much for your attention, and greetings for your sister city in Spain.